Year 11s, we're going to be looking today at what is alive and what is not. So we're comparing characteristics of living organisms to characteristics of non-living particles. One of the first things we need to consider when we're looking at whether or not something is alive and whether or not something is not is how we actually determine that. What are the factors that we use? And so we can start looking at this or examining this by asking ourselves a few questions. What is it that we consider is alive? Are plants alive? Animals? Are viruses? Are bacteria alive? And then the most important question of all is why? What actually makes them alive? When scientists ask these questions or ask themselves these questions, what they end up doing is creating a set of criteria. And these criteria actually help us to fit together all of these different whys. So we're looking at, we can use an acronym, which we use here is Mrs. C. Grant. So the M stands for movement, the R stands for respiration, S for sensitivity, C for control, G for growth, R for reproduction, E for excretion, and N for nutrients. What you need to be able to do with this is actually use it in such a way that you're going to be able to determine based on these factors whether something is or isn't alive. There are other acronyms or there are other examples, but they all come down to these few things. Now, sometimes you might find that a particular cell is stationary in a multicellular organism, so an example in our bodies are bones, but because we're considering the whole organism, we still consider that cell to be alive. When we're looking at unicellular organisms, they need to meet all of these things. So we're talking about single-celled organisms, they need to meet all of these features. And if they're not able to, then we won't be considering them to be alive. Where we're looking at investigation of cells, so how do we actually, what tools can we use, what skills can we develop to actually look at these particular questions, look at these particular situations, and determine whether or not an organism or a particle meets any of these or all of these requirements. And so the most important tool in microbiology are microscopes. And there are two different types. A light microscope, which uses visible light that we have around us, and then there's an electron microscope, which are microscopes that use far smaller particles or far smaller waves called electron waves, electron microscopes. They're able to see in much greater detail. Light microscopes allow us to see magnifications up to approximately 1500 times, and past that we have to start manipulating the light using a method called a synchrotron, or a device called a synchrotron. And you can look into that, and I recommend you do that in the internet. But then we've got the electron microscope, and theoretically we've got a magnification with an electron microscope of 1,000 times more powerful than the light microscopes. So we're looking at from 1,500 times up to 1.5 million times. Now in reality we've gotten to about 1 million times, and this has allowed us to see things in incredibly minute detail. We've been able to see not necessarily individual atoms, but sheets of atoms, and we've been able to create quite interesting things, these nanoparticles that we've been able to, or we've started to look at. There's some research going into some different types of new microscopes, and there's been limited success with those so far. So this brings us to this idea of a cell theory. If things are considered alive, what's a unifying thing that makes all of those things? Something that we might be able to see before we can examine whether or not it fits all of those movements and reproduction and all that sort of thing. What's our first port of call? And so this has given rise to this idea of the cell theory, which is basically three statements, three unifying statements: that all organisms are composed of at least one cell, that all cells came from a previously existing cell, and that the cell is the smallest organizational subunit of a living organism. That last one, about the smallest organizational subunit, is really just a statement saying that there is nothing smaller than a cell that can be considered alive by itself. There are no substances or no organisms, no particles that we can consider alive that are smaller than a cell, that don't have all of the necessary particles to be defined as a cell. And when we look at something like infectious diseases, we can see that there are lots of things that behave as if they're cells. Viruses and proteins are probably the most important two, or prions they're called, but they are not considered alive, even though they have a number of different factors that might make them alive, because they are not made up of cells. So we have these two main features that we're looking at when we're considering whether something is alive or not. Those 7 features: movement, respiration, sensitivity, what was C? Control, growth, reproduction, excretion, and nutrients. So we've got these 7 statements, or these 8 statements, sorry. And then we also have the cell theory, which is our discussion around that it has to be made of cells, the smallest organizational unit is a cell, and that all cells came from previously existing cells. In our next video, we're going to be looking at comparisons of different types of cells, prokaryotes and eukaryotes, and we'll break that into one type of cell with prokaryotes and then the second with eukaryotes.